So cancer this time, it's personal. It's personal, obviously, for everybody who's afflicted. And it's a pleasure for me to be here to talk in New Zealand, which certainly is one of my very favourite countries to visit. In terms of conflicts of interest, it's important to recognise these. I have advised companies about design of trials. We don't actually get many trials now without working with companies. I have chaired some of those trials, but I've always refrained from taking personal remuneration from companies. So where am I from? Well, I didn't have a big enough screen to put the countries together, but there's Canada and there's New Zealand. Not exactly to scale. New Zealand's actually a bit smaller than that. So a few things. Here's Toronto, which is where I've spent all of my professional life. Here's our flag. Now, one thing that's different is we didn't spend $27 million not to change our flag. We know this country is Lower Canada, although we might change that with the current way that the politics are going there. We're actually worried about influx of refugees in the future. And our connections with New Zealand have been quite numerous. I've been here 4 or 5 times, and my wife, who's with me today, Rosemary, has taught at Christchurch several times in the university, was actually here during the large earthquake that rocked that city, which was a very difficult time. So that's all for introduction. What I'm going to try and talk about today, give you an outline of the presentation, is to talk a little bit about mainstream cancer treatments that we have, talk a little bit about what is personalised medicine, about gene sequencing, about immunotherapy, and the problems, and there still are problems, and the likely benefits, and there are certainly many of those too. I'd like to talk a little bit about other research with potential to improve treatments, some of which I've been involved in, some of which people here have been involved in. A little bit on understanding clinical trials and thus the benefits and problems there. Obviously, a very key treatment is, can we afford these new cancer treatments? And finally, looking a little bit to the future. So let's start with today's mainstream cancer treatments. The cancer is a disease that starts off with cells, small cells, dividing, growing a lump, and initially that lump is in one place. So you may have a breast cancer that's growing in the breast, or a prostate cancer growing in the prostate in men. And the primary treatment for those lumps is either surgery or radiotherapy. And as your chair said earlier, radiotherapy has now become a highly sophisticated type of treatment that can use very sophisticated machines to direct a beam of radiation that can kill cells and spare normal cells, sometimes by rotating that beam around the body and focus it on the tumor. And I think most of you are obviously familiar with what surgeons can now achieve. But the big problem with cancer, why cancer is still a lethal disease for many people, is the problem of cancer spread or metastasis. So in our normal organs of our body, our cells know that they stay there. A breast cell stays in the breast, a stomach cell stays in the stomach, but cancer cells, through a variety of genetic and other changes, somehow learn that they can escape from one part of the body and go to another part of the body. And they can do that through traveling through the blood vessels that we all have, and they can go to other organs and there form a new lump. Now, if a cancer remains localized, and some cancers, when they are diagnosed, remain perfectly localized, most of those cancers, not all of them, but most of those can be treated completely and effectively by surgery or radiotherapy or both. But once the cancer has spread, and if it's spread to numerous parts of the body, then we need other treatments to approach that. And the treatment for cancer that has spread has to be use of other modalities that also spread where the cancer goes, so anti-cancer drugs. And there are a variety of anti-cancer drugs. Chemotherapy is a group of drugs that has been developed over the last 60 or 70 years. They are relatively non-specific in killing cancer cells, and we found by really trial and error which drugs work for which tumours. Chemotherapy is still by far the most common form of drug treatment that's used, and it is quite effective in many types of cancer. It is certainly part of the treatment that leads to improved survival in diseases like breast cancer, ovarian cancer, and so on. Hormonal therapy is relatively restricted to tumours or cancers that are growing in organs of the body that themselves are influenced by hormones. The breast, for example, gets bigger during pregnancy, shrinks later, under hormonal control. The prostate in men is stimulated by male hormones, so hormonal therapy for those diseases. And then we have this new concept of molecular targeted therapy, which I'll talk about in a minute, which is where most of the research is now going on to try to develop drugs that are more specific in attacking a target. As we've learned more about how tumors behave, about their molecular structure, people have been able to devise drugs, and newer drugs are being devised all the time, including some of them here in the Auckland Cancer Centre. And finally, immunotherapy, which has received a lot of publicity lately. It in part is, if you like, molecular targeted therapy, but it's a little bit different in that it seeks to activate the body's own defenses rather than to act directly against the cells that constitute a cancer. So what is molecular targeted therapy? So maybe a little bit more complicated slide. So what this is showing, and this is blown up thousands and thousands of times. So you have a cancer cell, and inside that cancer cell, which is, you could see with a good microscope, what happens is that there are various influences on that cell. They can be hormones, or they can be other factors that interact with a molecule that sits in the surface of the cell that we call a receptor. And that leads to signals inside that cancer cells. These are molecular signals, so these molecules are far too small to be seen on any microscope, so they are inferred by very clever scientific methods that allow you to work out the pathways, the molecules that influence the cell. And what those, in a cancer cell, those molecules, those multiple signals can activate cellular responses, and the responses divide and conquer. Basically, they're telling the cancer cell, you need to proliferate, to form new cells, to grow new cancer lumps, and to spread to other parts of the body. And molecular targeted therapy is where drugs have been developed, recognizing those signals that tell a cancer, and the molecular target agents, they inhibit those signals, they try to stop those signals that are telling the cells to survive, to divide, to invade and spread. So they're trying to stop those processes that allow a cancer to grow and, if it isn't treated, to kill the person that's growing it. So advances in science have led to drugs, designer drugs that are designed to inhibit specific molecular signals that are taking place in cells and telling them to grow and divide. And other active drugs also, which we'll come to. Now, there are downsides of these treatments. The drugs have side effects. These signals that take place in a cancer cell and tell them to divide are only a little bit different to some of the signals that are taking place in the normal cells of our body. We have to, even though we are adults, we still have cells in our body that have to divide and replenish cells that are lost. Inside our bones, we are remaking our blood all the time. Inside our intestine, we're replacing cells that are sloughed from the intestine. So those signals are not very different, and some of the side effects of those drugs occur because the signals are only a little bit different. And the other problem is that cancers are very clever. They can rapidly develop resistance to most of the drugs that we develop to act against them. And that is a big problem. It's been the limiting factor with chemotherapy and also with most of the molecular targeted agents. So what is personalized cancer medicine? How does this differ from the way we've been treating cancer over most of the last several decades? So if we have to understand that, we have to recognize that we have in cells genes, and the genetic material, DNA, is made up of an alphabet of 4 letters. A, T, C, and G. That's all we have. We don't have 26. We only have 4 letters. And the sequence of those letters in the DNA tells the cell to make a certain type of molecule, a protein, that gives it its characteristics. Each gene contains about 30,000 a sequence of about 30,000 of those primary 4-letter alphabet. And so that characterizes the gene. And we have in each cell about 30,000 genes. So it's a hugely complex process that science has unraveled over the last few years, characterizing the sequence of these genes. And it's become possible now with modern technology to work out the sequence of individual genes that do individual things in the cell. So, and if we can sequence those genes, and if we can do it for a tumor, a cancer cell, and if we can do it for a normal cell, we can actually see where the differences are. We can see how a cancer is different from the normal cells from which it arrives. That difference is called a mutation, a change. And a change in the tumor gene leads to a change in the protein, and it leads to a change in the way that a cancer cell behaves. And it enables us to design drugs to attack the pathways that are set up by that mutation. So if we can identify the mutations that signal a cancer to grow, then it should be possible to design a drug to inhibit those signals. And one of the examples is that women with breast cancer, about 20% of them, the breast cancers have a molecule that is known by this funny formula HER2. And I think many of you are familiar with the drug Herceptin, or trastuzumab, which has been around now for about 7 or 8 years. And use of Herceptin in women who have that HER2 change in their cancer cells has led to a marked improvement in the survival and cure rate for women who have that type of breast cancer. So this is where you do a test on the tumor. That drug has no effect whatever on the 80% of women who don't have that change or mutation, but it has a very big effect on those that do. So it's becoming, we're looking beyond breast cancer to now look at subgroups of breast cancer. And the same applies to many other types of cancer. And the idea of personalized medicine is that we might actually be able to extend this idea to identify changes that are in the individual tumors. Herceptin and HER2 identifies a group of patients, about 20% of women with breast cancer. Could we extend this to individuals, because every individual has some different changes within their tumors? And that's the idea of personalized medicine. So knowing molecular targets would avoid us trying to treat everyone in the same way. We know that if we treat a group of people with a given type of disease, let's say now ovarian cancer, that some of those women will respond to treatment, their tumors will shrink, in general about half of them in that disease, but the other half don't. But they still have the side effects of the treatment. So the idea here is, can we do tests which will enable us to determine which are going to respond to a particular type of treatment? Now, there are problems with personalized medicine. It's in its infancy, and it's not in the here and now in terms of individual treatments. And the problems are that cancer cells often have many mutations, many changes, 30 or 40 perhaps. Which ones are important? And which ones are actually driving the cancer cell to behave in this abnormal way versus being what's called passengers, where they're simply present but not really influencing the way the cancer is behaving? That's something that the scientists are trying to work out, and they have a reasonable handle on that. Resistance is a big problem. If we inhibit one mutation, we may get a response in the tumor, but the problem is that the cancers, as I said, they're quite clever in learning to overcome that problem. They tend to change their behavior, and that is one of the problems. We need more than one drug, we need several drugs, and sometimes it's difficult to put the drugs together. Different parts of tumors can have different mutations. So if we take a biopsy from one point, which a surgeon can easily do, or even in the radiology department now, you may get a sample of a cancer that's representative of that sample, but there may be other parts that are not quite the same, so you need a different approach. And the drugs currently available have still many side effects. Now, all of these are changing. All of these, there is research going on to try to improve the outcome in all these different aspects, but there's a lot of work still to do. One way of looking at it is like this. This is a tree. So here's an imaginary tree, and unlike most of the trees, it's got different colored leaves. And what those leaves represent are parts of the cancer that have different properties. So within a cancer, you can think of a cancer growing from originally one or two or a small number of cells, and as it develops, then different parts of the cancer may have different properties, and that's represented by the different colored leaves there. So you might have a good drug that's good against the leaves that are reddish or greenish or purple or yellow, and you could use that drug, and after treatment you will have usually some shrinkage of the tumor, and it may be a time before that tumor grows again, but as long as you still have these other parts of the tumor that weren't responsive to that drug, you will need other drugs to treat it. So it is a problem of these differences in different parts of the tumor. So let's come to immunotherapy. So what is immunotherapy? Well, we've known a lot about the immune system for a long time. You all know that when, most of you probably have children, your children get sick, they get a virus, they get a bacterium, and they get better. Thank goodness. Why do they get better? Because they have an immune system that recognizes foreign molecules, foreign proteins. The technical term is antigens, and it acts to destroy them. So we have two types of cell, the police force in the body, if you like, B cells that make antibodies, and T cells that directly kill foreign bodies like bacteria or viruses, particularly viruses there. So we have an immune system. And for about 100 years now, people have had the idea that, well, tumors are a bit different, so can't we somehow get the immune system to get rid of the tumor? There are a couple of problems there. First of all, the tumor has grown despite the immune system, whereas generally viruses and bacteria don't continue to grow in the body. So obviously it's got ways of circumventing the immune system. And secondly, the differences between cancers and your normal organs is actually very small, whereas the difference between a bacteria in your normal organs or a virus in your normal organs is quite big. So it's a big problem. And for many years there were immune treatments, and very occasionally you'd see some pretty dramatic results of using those treatments, but it wasn't consistent. Other people would have no effects at all. So potentially the immune system can act against tumors. And as I say, this idea is put forward for decades, but it's only really recently, in the last decade, even less, that immunotherapy has shown consistent benefits for people with some types of cancer. And how does modern immunotherapy work? Well, what they've discovered is here is an imaginary tumor cell, this sort of very folded-looking cell with a nucleus, and this is something that you'd be able to see with a good light microscope. And here is an immune cell, we call it a T cell, that potentially could bind to it, but the rejection of that tumor is turned off. And what we've found is there's a lot of signals that turn off the immune response. And the recent advent of new drugs like pembrolizumab or Keytruda or nivolumab or ipilimumab is that these new immunotherapy drugs, they release the blocks, they take off the brakes on the immune system that in some cases now can have an effect to really treat the tumor. And so that's been the big change. Most of that science has been developed in publicly supported and charity-supported laboratories, but now those drugs have been developed for use in the clinic. And this is showing one of the results. This is an article by Caroline Robert, who's in Paris, and colleagues. And just to explain what we're looking at here, this is a clinical result of a large clinical trial where a large number of patients, several hundred, and they had some melanoma, a common disease in New Zealand, a skin tumor that has spread. And they, by random choice, were randomly accepted to receive the old treatment, which is chemotherapy, which was not very effective. And this is the proportion of patients who were surviving at different times after treatment. And you can see that with the old treatment, by about 15 months, most of the patients had succumbed to their tumor and died. But with the new treatment, many patients are now having, at 18 months, they're still alive, and the disease is not progressing. So the very interesting thing about these new drugs is that they don't just improve the average survival, but there actually seems to be a proportion of patients, it won't remain at 70%, it'll come down from that, but a proportion of patients, maybe in the range of 30 to 40% with these new drugs, that may remain well for a very long period of time. We don't yet know if those patients are completely cured, but they can remain well for a long period of time. That is an incredible advance in terms of treatment of that disease. Now, these drugs are also being looked at in other types of cancer. They have shown smaller effects, not effects to prolong survival in the same way as in melanoma, smaller effects in diseases like lung cancer and in kidney cancer, and there are other trials going on in other diseases. So the next few years are going to be very exciting to see in which diseases and by how much these drugs can improve the outcome of people with cancer. But melanoma is the disease for which it's happened so well. Is immunotherapy personalized medicine? Well, not really yet, because drugs like pembrolizumab and nivolumab, they target a particular target on the cells and release the brakes on the immune system. But up to now, we've used them, or the people who've done the trials have used them in all of the patients with a given type of cancer, such as melanoma. We haven't yet selected out the patients. People are putting a lot of effort to try and find, is there some marker on the cells that will separate out those people, maybe close to half of them, that have a profound response from those that don't, because obviously they're obscenely expensive drugs, and we'd really like to be able to use them wisely to treat the people who really need them. But immunotherapy could be adapted to overcome the problems of this variation that I told you about. Remember the tree that I showed you? Well, there's some recent work come out from a laboratory in London showing that if you can have immunotherapy that is recognizing differences in the trunk of the tree that remain there, then it may not matter so much that the leaves are different. We don't know, but that may be the case. Just a cartoon, though. I'm afraid neither your insurance nor your immune system will cover it. And I'm well aware of the payment problems, even in wealthy countries like yours and mine. I want to touch on other examples of translational research. You know, there's a lot of work, appropriately, there's a lot of research going into personalized medicine, there's a lot of research going into immunotherapy, but I think history teaches us that it's a mistake to put all your eggs in one basket. We will need, in different types of cancer, different approaches to trying to treat the disease. And one of them, as I've already mentioned, is that most cancers cannot be cured by drugs because they develop resistance. And possible causes of that are the cancer cells themselves are resistant to the drugs, and that's a big problem. But another problem, which has received a fair bit of attention here in Auckland, but not so much worldwide, is that often our drugs are not very effective in getting to all the cancer cells that need to be treated. And I gave a research seminar earlier today, and I showed this slide. And what it is, you'll have to take my word for it, is that looking under a microscope, it's a picture of a liver of a mouse. And this mouse has it's a mouse that will accept growth of a human cancer, and that cancer is spread to its liver. And this is actually a picture of its liver. And those big black holes there, where these arrows are where the cancer has formed metastases, has spread to the liver of that mouse. And these rather bluish cyan-colored areas are the normal liver. And why are they cyan? Well, in fact, what we did with this mouse is we gave it an anti-cancer drug, a chemotherapy drug, that is fluorescent. So when you look at it under the microscope, you can see it. It lights up blue. And what you would notice is you've got tons of drug in the normal part, the normal liver that you don't want to treat. There is drug in here. You can, if you're very close to the screen, you can see little bits of blue, but there isn't very much. And one of the problems is that cancers don't have a very good blood supply. May be positive in some ways because it will slow down their growth, but it also means that when you try to deliver drugs through the blood supply, then they're not getting in very well, and that's a problem. And in my lab, and also in Bill Wilson's lab here, we've studied some of these problems of drug delivery. And this is a recent graduate student of mine, Jas Sagar, who did some work looking at how chemotherapy damages cells in tumor. And she looked, this is a control tumor, these red things are blood vessels, they're stained. And here is markers of damage after we've treated chemotherapy, this one blue and this one in yellow. So you can see markers of damage, and we can trace those markers of damage as we go away from the blood vessels. And as you go away from the blood vessels, the amount of damage, which is represented by this scale, falls off as you get further away from blood vessels. So when you get out here, where there's a lot of areas of low oxygen in the microenvironment of the tumor, then that damage is falling off. And so one way that chemotherapy may be failing is that we give chemotherapy that comes into a tumor, and this is a cartoon representing the inside of a tumor. Here's a blood vessel, here are cells, these would be seen on a microscope. The cells that have a lot to eat, they have nutrients coming from the blood vessel, are dividing. As you get further away, they tend not to, and their cells are stressed, they don't have a lot of nutrients, but they can still survive. And what we believe is happening is that with chemotherapy, if the cells are sensitive, we can get rid of those cells, but unfortunately what happens, those further away divide, and then they re-establish that structure. So what we really need is to be able to attack those dark blue cells. I went to Cambridge, which is light blue, and the dark blue is called Oxford blue. We need to attack Oxford, that's what we need to do then. So, if cancer cells far away from blood vessels survive and regrow the tumor, what can we do to attack those cancer cells? And there are a couple of strategies that we've used. One is to inhibit a process called autophagy, and you can all sit there and say, well, what is autophagy? I'll tell you in a moment. And secondly, because those cells, those dark blue cells further away, tend to lack oxygen, we can use drugs that might be activated inside those cells. And that's where the Auckland Cancer Research Centre is doing a lot of research. PR104 was mentioned. It is a drug that attacks cells by being activated under conditions of low oxygen. So what is autophagy? Autophagy means self-eating. And so what it does, it breaks down damaged components of stressed cells. And it can be a survival mechanism for stressed cells. It's your recycle box. It allows recycling of cellular constituents. And autophagy can be inhibited by some drugs used for other purposes. Chloroquine is an anti-malarial. We've got anti-ulcer drugs such as pantoprazole. I'm sure many people in this room take low doses of pantoprazole. So let's have a look at it. So that's how we recycle in Toronto. We're very simple. We put our glassware and our papers in a blue box. You're much more sophisticated here. You've got lots of recycling boxes. So this is the process of autophagy. Here's another of my recent students, Susie Tan, who did some work. And the only thing you need to know in looking at these pretty pictures is that red means you've got lots of autophagy, and green means it's inhibited. So lots of autophagy means the cells are stressed, they're using the mechanism to try and survive, to try and overcome what we're doing. And along the top here, these things are initials of 8 different commonly used anti-cancer drugs. They include drugs like gemcitabine, docetaxel, doxorubicin, which is adriamycin, etc. And you can see that each of these drugs stresses the cells, and they use autophagy, and we believe they use autophagy to try and divide. And we also believe that's a particular problem in the parts of the tumor where conventional drugs aren't getting. And then we can use high-dose pantoprazole, that same drug that many people take for stomach upset, and it turns them green. Now, it's easy to do that in culture. That means we're inhibiting autophagy, and we can hopefully get better effects against tumours. And indeed, we do find that. So when we look at tumours growing in mice, these are different human tumour cell lines growing in mice. This is a measure of tumour size. This is a measure of time when it's growing in the animals. These lines represent no treatment, or the anti-cancer drug alone, or pantoprazole alone, but the red curve where we're stopping the growth of the tumour is where the drugs are used together. And we see that for 3 different types of tumours there. And we've actually taken that into some clinical trials. My belief is that we need better drugs. We need better drugs to inhibit autophagy, and indeed some research centres and some small companies are now trying to develop drugs like that. So what I see in 2025 is that there will be greater recognition of the problem of drug delivery within cancers. That's already happening, actually, in many of the companies. I think new drugs will include inhibitors of autophagy and drugs activated under hypoxic conditions. Moving on, understanding clinical trials. Now, it's one thing to develop a drug in a laboratory. It's one thing to show that it acts to treat tumours in mice. If we could do as well in people as we do in mice, we'd be a lot further forward than that 57% that John mentioned at the beginning. And I think it's very important for people to understand at least a little bit about how a drug goes from a laboratory, like the laboratory just here, into clinical trials. So here's a drug. The first thing you have to do with a drug is give it a good name. So with this drug, a hypothetical drug, I called it Miraculin. And what you might find is the drug causes death of cells in tissue culture. It inhibits growth of tumours in mice, much as I've shown you, and the maximum tolerated dose is determined usually in different animals, and then it goes into a series of clinical trials. The animal work helps you choose the dose. And we clinicians are pretty logical, so we call our first trial a phase 1 trial. And obviously when you're first taking a new drug into people, you've got to be very careful. You've got to start at a low dose. You have to give it to volunteers with advanced cancer, and you really need to give it to people where all the conventional types of drug have already been used. It would not be ethical to give a brand new drug, however promising in an animal, to a person who had not benefited from acceptable treatment. But there are many people like that, and we do a large number of phase 1 trials in the institution I come from in Toronto. The main aim of a phase 1 trial is to try to find a good schedule, to try to find the right dose, and to look for side effects. Then, if the drug seems to be behaving itself okay, that the side effects are tolerable, hopefully you see 1 or 2 patients having at least benefit in terms of tumour shrinkage, we then do small trials, usually in groups of patients with the similar types of disease. So we might treat a group of patients with melanoma or with bladder cancer, and increasingly we're selecting those patients not only on the basis of the type of cancer, but also the type of mutations that they may have. And then finally, if we see good effects to shrink tumours, that doesn't prove that you're actually going to improve the survival, the life expectancy. You need to do large trials where you compare the drug with a standard treatment. And some of you may have come across this strange thing of randomisation. So you want to test a new drug. I showed you the example of the immunotherapy drug. So you want to compare it with standard treatment. Now, if the doctor chooses which patients get the new drug, there will be a bias to who he or she may think may respond. Likewise, if a patient chooses. So generally speaking, patients accept in that trial to have either treatment based on randomization, if you like, by the toss of a coin, although it's done with computers. It removes bias. It means that the patient characteristics in the 2 arms of the trial are similar, And sometimes we even do that so the patient and the doctor don't know which treatment is given. The pharmacist makes it up, the patient gets the treatment, and that removes all sorts of bias. And what do we look for? Well, the outcome measure should reflect patient benefit, ideally how long people live, or the quality of the survival. And we need to look more at quality of survival. You need large trials of several hundred patients, and ideally we need to include people who are representative of those we see in the clinics. One of the problems of clinical trials, particularly those sponsored by companies, is a tendency to select otherwise very healthy patients with cancer. And obviously then you get it will make the results often look better, but when we then come to treat in the clinic, cancer often affects older people who may have other problems like hypertension, high blood pressure, cardiac disease, etc., and they may not get into the trial. I'm going to show you a couple of examples from prostate cancer, which is a disease that I've been involved with developing treatments. And the previous treatment options for many years were to remove the source of male hormones, either by removing the testicles or more recently by using drugs. Men don't like the former very much, so they prefer the latter. Research over the last few years, followed by clinical trials, has led to two new drugs, abiraterone, which is known as Zytiga, and enzalutamide, which is known as Xtandi. And abiraterone is available here for people in New Zealand. And here was the clinical trial that first established the abiraterone. So again, we were looking at men with prostate cancer of advanced stage, spread disease, already had most treatments, and they either had a placebo or abiraterone. And you can see that when you look at those surviving as a function of time, it's higher on the drug. So at a year, about 60% are surviving here, about 40% who didn't get the new drug, and enzalutamide was much the same. And we're also learning about research, and you're probably looking at this sort of crooked fence that maybe to keep horses into a field and asking what on earth it is. Well, it's actually where science can show what a molecule looks like. And I mention male hormones because the way these drugs work is by decreasing the effect of male hormones on a molecule, and we call it the androgen receptor. Androgen's a male hormone, so this is a molecule. It's like you've got a lock, you've got a hormone comes along, puts the key in the lock, turns it, and then turns on a process. So this it doesn't look like a lock, but that's what it is. And for it to work, it's got to have this green ending here. LBD stands for ligand binding domain. What that means in simple English is that's where the stimulating molecule comes along, interacts there, and tells the prostate cancer cells to divide if it's the male hormone, or if it's the inhibitor, to tell it to stop dividing. And what people have found is that when they become resistant to these drugs, they lose that. It drops off. The molecule gets rid of it. It's part of the way, as I told you before, that cancers adapt and become resistant. So they lack that part. And what people found, this is a group in Johns Hopkins University in Baltimore in the United States, is that if they lost that part, then these are the proportion of patients who don't have progressive disease, that drops off very quickly. Everybody has progressive disease in 3 months, whereas they have the normal receptor, they last much longer. So how science can help to overcome resistance, and what I will foresee in prostate cancer in 2025 is that whereas these drugs are now very expensive, they'll get a lot cheaper because generic compounds will be made. The companies fight those, but they will be made. We'll understand causes of resistance to the current drugs, much as along the lines of the research I just showed you, and we'll develop actually new drugs that will target pathways that avoid the resistance. We'll develop drugs that act at different parts of that squirrely molecule that I showed you. And we'll need innovative trials to define the best treatments. But clinical trials do have some problems. They are designed by drug companies. Drug companies are not all bad people at all. They do a lot of good development, but making a profit and giving the best treatment for patients is not always they're not always going together. And sometimes there are biased reporting and selection of patients. And one of my previous fellows, Paco, he's a Mexican, looked at biased reporting of trials in breast cancer, and there were 164 large trials published in that time period, and about a third of them showed benefit of a new treatment, about 2/3 didn't. And that's fine. I mean, it's not a bad batting average, but unfortunately in the reporting of those trials, sometimes secondary outcomes were used to suggest benefit, and there was a tendency to underestimate the toxicity. And we really do need to try to improve the reporting of the clinical trials, which is a particular interest that I have. And the other thing we need to do, it's not enough just to do clinical trials on a very selected population. As I already said, sometimes we select out very fit patients, otherwise fit patients who have cancer. One of my colleagues once said you have to be an Olympic athlete with cancer to get into a trial. It's not quite that bad, but we do tend to reject people who have other problems. And clinical trials show treatment effects in people selected most likely to benefit. So we have to ask, do these benefits persist in a general clinical practice? And one of the things, this is a colleague of mine, Chris Booth, who's now at the National Cancer Institute of Canada Research Centre in Kingston, Ontario, but trained with myself and others. And he and others are using the Ontario Cancer Registry, which is a very good registry. And we now have computer methods so we can link databases in hospitals to the registry, and we can actually ask, when we have a new drug, is it affecting the whole population, not just those in the trial? A very good drug, it almost always will. But some of the more marginal drugs, then you may lose the effect. And you can see the differences between clinical trials. So another fellow with me, Arnu Templeton, who's back in Switzerland. And if we looked at using chemotherapy for men with prostate cancer, a trial that I chaired, if we compared the patients on the trial with those that we've treated routinely in our hospital, then you can see that the average life expectancy is lower in routine practice, largely because people have other medical problems and side effects are higher. So we do have to be careful, particularly with more toxic drugs, in applying the results of trials. We're coming to the end. Can we afford the new treatments? Well, it's difficult. So this is the average cost. So you can measure the effectiveness of a cancer drug by the average number of life years that it improves it gives you compared to the older treatment. And as John said at the beginning, that's happened. We have much better drugs than we did 50 or 60 years ago, but at the same time, the cost for saving a year of life has gone up dramatically. As you can see here, from 1996, around $60,000 for a life year saved, to now well over $200,000. And even in wealthy countries, that's a huge problem. Do you spend your money on improving cancer treatment? And if so, where do you put it? Or do you spend it on enhancing children's education or any other thing? So it is a problem. And people are taking this to heart. I show this for amusement. This is Le Figaro, which is one of the major national newspapers in France. I happen to know these two gentlemen you see in the picture. And there, virtually all the leading oncologists in France have signed a petition to try to force the companies to reduce the cost of the anti-cancer drugs that are being produced. And I know that's very much in the public eye here in New Zealand. And clearly there needs to be action on the basis of concerted action from physicians, from patients, from politicians. It's difficult because the price is largely set by what happens in the United States. So many people are denied effective treatment, not only in countries like ours, where I think most of our patients get pretty effective treatment. But even more, we're paying equally high prices for some rather ineffective drugs where we're buying a Ford, but sometimes we're paying for a Ferrari. And so we need to use that drug expenditure, I think, much more wisely. And some of the new immunotherapy drugs are certainly Ferraris, but some of the older drugs give really rather marginal benefit, and I think we could reassign some of that cost. So looking to the future, I think we're going to see continued research into the causes and properties of cancer. In fact, I know we are. And regardless of that having an immediate effect on therapy or not, and I think it's probably mostly a little bit down the road, it's going to teach us more about the disease, and it's the way that advances have taken place in the past. We are going to have more effective treatments. Immunotherapy is the most exciting that is out there at the present time. And it'll be very fascinating to see when we'll be using these drugs, not only for advanced disease, but after surgery, for example, in people at risk of developing spread of disease. And we don't know if they will work in that situation, but those trials will be very interesting. And personalized medicine, I'm a little bit more skeptical about the individualized personalized medicine for the reasons that I stated earlier. But nonetheless, we need to see if it will work. But we also need to make sure that the research that we're doing is diverse and not all put into that one pot. So that's the end, and these are acknowledgements. These are the people who worked in my lab. You may think that my colleagues are all young women, which is very nice. But these are actually two of my postdocs and a separate picture. And then in terms of the clinical sphere, some of these, I've shown you their work. These tend to be much more male, of the fellows with whom I've undertaken some of the studies. So thank you very much. It's been wonderful to be here, and good luck with your Cancer Week and with the future of your society.